Welcome to a new chapter of Glioblastoma, aka GBM, presented by the Glioblastoma Research Organization. This season, we're adopting a documentary-style format to offer you a never-before-seen look into the world of glioblastoma from around the globe. Join us as we venture into labs and companies worldwide, providing a comprehensive view of some of the cutting-edge efforts to combat GBM. Through intimate interviews and detailed on-site explorations, we'll offer an insider's perspective on the innovative strategies and real-time progress in the fight against glioblastoma. Thank you to our sponsors whose generous support has made this season possible. Please note that the sponsorship of this season does not constitute an endorsement of any sponsor's products or services. Remember, the information presented is for educational purposes only, and it is not intended to treat, diagnose, or prevent any disease. Always consult your medical provider and healthcare team with any questions.
First of all, my name is Saskia. I'm a medical doctor by education and specialized in human genetics. And in 2009, my husband Dirk and I, we founded CeGaT. And CeGaT is specialized in genetic testing. And we were one of the first companies to provide next generation sequencing to patients. And now this is what we do. We offer the most modern and most recent available technology to our patients. Many diseases, and you can look at epilepsies, muscular diseases, Parkinson's disease, dementia, skin diseases, heart diseases. It's now known that the genetic contribution to many diseases is huge. And the technology we use has the aim to identify the cause of the disease in the human genome. So if you think about that, one variant or one mutation in the genome can cause a severe disease, this is what we try to find. And when you have a patient, for example, with epilepsy, you try to find the cause in order to find treatment. And this is the same thing we do with patients with cancer. We try to understand the disease, we identify the mutations, and then we develop personalized treatment based on the genetic information. If you think about someone who is sick and has a specific health question, they have to undergo a lot of diagnostics. They get ultrasound, MRI, CT, blood tests done, but genetic testing is not part of this of these analyses. And the vision to found CeGaT, and still this is our vision, is that every patient should get access to genetic testing, because it's important to know or why a disease is occurring, to understand the basic or the underlying mechanism of a disease in order to then design treatment. So I think it's crucial that every patient gets access to genetic testing. But genetic testing is not available to everyone, because not everyone has access to the technology, and then also it's expensive. So when we started CeGaT, genetic testing was like 10 times more expensive, and we could only look at one gene after the other. With the technology we are using now, we can sequence hundreds and thousands of genes in parallel. Actually, the whole genome can be sequenced within days. And the costs went down dramatically, but still it's a lot of work and it's still expensive. So this is why one has to think the benefits of the genetic testing, they are much bigger than not doing genetic testing. If you, for example, do not find the cause and you need years to find the reason of a disease, then it's much more expensive than doing genetic testing in the first place. So we really fight to implement genetic testing in the first place for every patient. For all our patients, we use the latest available technology. So at the moment we use the Nova X Plus machine, and we have several of those, and there is no machine that is faster and better than this machine. We like to have competition, so we want other companies to provide new technologies, and once we identify a new technology, we will implement it. We need to have the best quality, we need to be fast, and we need to be cost efficient. And there are new technologies developing all the time. For example, spatial transcriptome sequencing is something that is very new. What does it mean? We not only have to understand the genome, we have to understand transcripts and proteins, because in glioblastoma, in the end, we need to understand what is causing the disease and why is the disease spreading in the brain, and what happens not only in the tumor but also in the microenvironment. So we also analyze immune cells. We want to understand how the tumor interacts with the brain, with the immune system, how the metabolism metabolism of these cells function? Why do tumor cells have advantages over immune cells, for example? And this we call omics technologies. We try to understand a lot around the tumor, and from this we have to draw the right conclusions, which is really challenging. And you will speak to our head of bioinformatics later. So our expertise is not only to generate the data, but also to understand the data, to analyze them in the right way, and to give a recommendation to a patient. I think it was completely underestimated that variants in genes play a role in the disease pathogenesis. Previously, it was thought that a glioblastoma is not inherited, but there is a fraction of patients where we really see that this can be an inherited disease. And this is relevant because based on this information, you can develop treatment strategies. Also, when you look at glioblastoma, it's not like this is one disease. Every patient has a different disease. So meaning that what has been done the past 20 years is they have all been treated the same, which doesn't make any sense. So what we bring in from a genetics perspective is that every disease is unique, and this is why you cannot predict the outcome. And so it's not one disease, it's like everyone has its own disease. And this requires that we analyze the disease in very detail in order to come with a fully individualized strategy. And a personalized or individualized vaccine is fully designed to attack one type of tumor and not another one. And so what we bring in is basically the uniqueness of a patient and the disease, and then also based on that, the uniqueness of the treatment. And this is why we hope that with this we can move one step further and get ahead of the disease at some point. A patient gets diagnosed with a disease, and then this is a very overwhelming situation, and then it's very difficult for the patient and the family to figure out what options there are. Like I said before, genetic testing has to be done as a very first thing. So if a patient gets connected to us, we tell the patient what we need, and we need a blood sample and a tumor sample. And this is very simple. You can ship a blood sample and a tumor sample very easily at room temperature. It doesn't require any specific conditions. Once we receive the blood and the tumor, we will prepare for sequencing. And then we analyze both tissues in parallel. And you will also see our sequencing machines. So the samples will go on the sequencing machine, and a lot of data will come from the machine. And then our bioinformaticians, they prepare the data for me or for our biologists so that we can analyze them. And then what we do, we identify the variants that are unique to the tumor. We want to understand the differences between the tumor and the normal cell. And then we write a report. And this report contains what is driving the tumor, but also the mutations that are relevant for a personalized vaccine. And this data we bring in an interdisciplinary independent board because we want to review the situation of a patient and understand whether there are additional treatment options. The board is then making a recommendation, and we work with experienced centers in the US. They don't require our board, but they get our data and they implement it into their treatment strategy. When a personalized vaccine is recommended by the board, then it comes back to us because we then manufacture the vaccine. What we generate with our report is basically like a recipe. It contains what we call peptides. They cover the mutations of the tumor, and we can synthesize them. And we manufacturing the vaccine from the scratch for each patient, and then we bring it into a little vial, and then basically after 12 to 14 weeks we have a fully personalized vaccine on the table waiting for a patient. And then the patient gets appointments with us, comes here, and then receives the personalized vaccine. We have to see this as a really big teamwork. So we built hopefully Champions League team around the patient, and we have usually an excellent neurosurgeon, we have the neuro-oncology team, we have the neuropathologist, we have the neuroradiologist, and they of course design the treatment strategy independent of what we do. And then we come on top and we provide the personalized vaccine. And so a patient can take basically everything. There's no exclusion criteria. But what we try to do at the moment, I think what is necessary is to do this within a clinical trial in order to get into the approval process. At the moment, patients have to travel, they have to pay, and they need to find this additional treatment options. But in the future, it should become standard of care that you are treated personalized. First, you have to do a clinical trial, and you have to show the evidence that the treatment is providing a benefit. And this can take years and costs a lot of money. If everything goes well and we can reproduce the data that we have already seen in real world, then we would aim for an FDA approval. And then it can become part of standard of care. I know from a genetics perspective that humans will always have cancer. So it is the way on how we approach the disease and how we treat it. And I think there's no place for chemotherapy in the future. We have to get away from these archaic and very destructive forms of therapies towards recognizing that the immune system plays a huge role, supporting the patient's immune system and providing access to immune therapies to every patient. I think these are the next steps. Genetic testing in Germany is mainly covered in a situation where you have a disease which most likely has a deadly course. You're running out of therapeutic options, and there's evidence that other therapeutic options could be a benefit for a patient and are not harming. So I'm actually spending quite some time for my patients that are from Germany in order to discuss with the insurance to cover the whole process. And we are having success for some patients, but not for all. In the US, it's more difficult for me, but our CeGaT testing is now also covered by some insurances, which is a big step, because then at least the diagnostic part can be taken by the insurance. I think that should be an aim, because diagnostic testing is crucial. There's no reason not to do it, so I think it has to be done. Very frequently we find additional treatment targets. So there's a subgroup of brain cancer which contains the IDH mutation. There's treatment available targeting IDH. For glioblastoma patients, we frequently find gene fusions, and there's treatment approved for other patients that can be used. And also very frequently we find variants that can be targeted by lung cancer medication. So genetic testing, I think, has to be covered by the insurance, although for many patients still we have to discuss this with the insurance. When it comes to treatment, targeted treatment that is approved for other cancer entities is hugely expensive. If you look at costs for inhibitors, you speak about thousands of dollars per month. Our treatment is not approved by the FDA, and that makes it challenging because there's no formal evidence and no approval. And so the insurances might say, we are not covering this because it's not approved. Yet we have some real-world evidence that we published last August, and we can discuss with the insurance. And then also when you say this treatment might be more beneficial, then potentially the insurance does not have to pay for a treatment that is potentially not beneficial but is costing a lot of money. So then you can say, we rather do this and leave the other. But, and now comes the very important thing for glioblastoma patients, there is no magic bullet. You always have to combine strategies. And the reason for comprehensive testing is to generate as much data as possible, to have the data for the interdisciplinary tumor board meeting, and then being able to suggest the right combination, because you don't want to have side effects. You want to be efficient in terms of costs, and you want the best outcome. And this is an interdisciplinary decision. And if the team comes up with a strategy, I think you can discuss this with the insurance. But you have to fight against the disease, you have to fight against the structures, the regulations. So for most patients, I think it's too overwhelming. And I think it's good to have knowledge, and this is what you do. You help educating, you need contacts, you need to know whom to talk to, how to build your team, to know who is primarily navigating through the whole process. And I think you and us, we help patients to find people to help navigate.
My name is Florian Battke. I'm working at CeGaT. I've been working here for 13 years and started in bioinformatics. I'm now Director of Development, which means that I oversee the development of our bioinformatic pipelines, but also of new projects. We have decided to keep every bit of our data in-house, so we're not using cloud services. On the one hand, it's for data protection. On the other hand, it just gives our customers a good feeling. So they really know that we have a handle on all the data and we're not giving it to anyone. And that means, of course, that we have to invest in infrastructure. So we set up a large storage system here in-house, which is also very secure. So it has several locations where the data is stored. It's a flash-based system, so it's also very fast. That's important for us. And then, of course, we have our own compute infrastructure for data processing. This is usually some regular servers, such as you could use for anything, and some bespoke servers just for processing of whole genome data. There are two main challenges. One is that the technology keeps evolving very quickly, so we cannot just set up one test, validate it, and then be happy for the next 25 years. We get new sequences every 3 to 4 years, and they completely change how the sequencing is done in the machine. And of course, also the knowledge on genetic diagnostic advances. So we have to introduce new analytes, or analyze is what we measure. So we look at sequence variants, copy number variants. Now we're also looking at structural variants, at viral infections. So there's new things coming that we need to validate. In the same moment, there aren't many data sets that are really well described. So if you want to do a validation, you have to process data, produce results, and then compare against a gold standard. And that doesn't exist for many of the things we're doing. So that is the challenge in validation. We have to do a lot of validations quickly within a price, and also there's sometimes the lack of gold standard data. So when I said there is new sequencers coming every 3 or 4 years, this means we get new machines that are faster, they produce more data in the same time. And sometimes also the quality of the sequences gets better, they get longer, and that enables more accurate analysis. This is not specific to GBM, of course, but better quality helps all our patients. Genetic diagnostics has basically two parts. One is the medical part and one is the technical part. The medical part is which genes do we need to look at and what can be learned from the variations we see in these genes. And this is, for example, for glioblastoma, there's a certain set of genes that we need to look at. It's pretty much the same as for all other tumors, but there are variants that are more common in glioblastoma. So that's the medical side. And this evolves over time with more knowledge being gained. On the other side, we have the technical development where the machines get faster, they get more accurate. So the one doesn't really influence the other. Now, for example, if a machine brings a new breakthrough, for example, now we have long-read sequencing machines that produce longer sequences. And these can be used to detect structural variants with a higher precision than with the short sequences we had before. So this new technology can now inform the medical side where we say, maybe in glioblastoma structural variants play a role. So I'm not a medical doctor, I don't know if that's the case. We can go back to our cohort of patients and maybe sequence some interesting cases with a new machine, learn something from that and maybe publish a paper on it. So the current development at CeGaT is again also split into the medical and the technical part. So for glioblastoma and other brain tumors, I'm not the person for the medical part. This is being done by biologists and medical doctors who think about what does the literature tell us what we should look at for glioblastoma in the future. And this might not just be genetic sequencing. We might look at the microenvironment of the tumor. We might look at certain bacteria that might play a role. This is the medical part. And then they come to me and say, this is what we need from the technical part. And then I'm developing on that. So as I mentioned, for example, structural variants is something we're working on right now. That's a technical solution to many questions, and one of them might come from the glioblastoma space, or it can come from the epilepsy space. I don't know that part. When we're looking at the development of cures for different cancers, there has been a lot of progress in the last, say, 40 years, right? So, for example, in leukemias, people would just have a death sentence handed to them, and now there's different medications that can make the leukemia just go away completely for certain cases. And for the brain tumor cases, there have been some developments, and again, I'm not a doctor, I cannot tell you exactly which ones are effective to what degree, but the important thing is to learn more about how these brain tumors develop and how they evade the medications that we have. And this is why we need to look at the genetics and at all these other factors that are involved. And I mean, researchers are really trying hard to understand this. It's also very hard because it's the brain. It's not as easy as a melanoma where you can just take a piece of skin and sequence it, right? I'm always, that's a bit of my personality, I'm always excited about what I'm working on. It could be different things. Right now in the diagnostic space, as I mentioned before, is the structural variation. This is something that with the current technologies, it's really not easy to get at. We get a lot of false positives. And as I mentioned earlier, the long read sequencers will bring or promise to bring some better resolution here. But then if you produce the data, you have no real reference to compare it to make decisions diagnostically, because we haven't seen large cohorts of patients really well characterized like that. So this is something that from the technical side is getting better. We're working on the bioinformatics side, and then the medical side also has to do something. On the other hand, there is in more general in genetics, there has been a discussion if you should do whole genome sequencing for everybody, maybe even just after birth. It seems like a simple answer. Yes, of course, this is the test you should do for everybody, but then the technical details are intricate, so sometimes it's not the best test to do. And these discussions, I find these very fascinating. One thing that I would be excited about, but that we haven't really found even a way where how we should get there, is to be earlier in detection. Especially with glioblastoma and brain cancers, brain tumors. You don't feel them, you don't see them, they don't grow a lump on your body somewhere, and then suddenly you start having loss of function in your brain, and then it's too late already. So this is something that I would be super excited about for that. Even genetically, for example, for other tumors, it's easier. They shed pieces of their DNA to the bloodstream, so you might be able to pick them up with some early testing. For the brain tumors, this is a rare event, so it's really hard to find them without looking at the brain. I mean, everybody has friends that have cancer, everybody has also lost people to cancer, and oftentimes it's just a case of late detection or some missing treatments, as you mentioned earlier. But this early detection would be something. I mean, you could have cancer, I could have cancer right now, sitting here, I don't know about it. It's a weird feeling, making yourself aware that, I mean, you have cancers growing right this moment. Most of them are going to be fought by your immune system successfully, so you'll never know, but one of them might not. So if we get this early detection sorted as a species, that would be great.
My hope is that we completely rethink the disease. That starts with understanding, and CeGaT is primarily contributing to understand each individual disease. Not to fall into the whole statistics and getting bad news, but first trying to understand what each patient has, finding the molecular markers, understanding where the situation is exactly, and then coming up with a plan. We are not saying that we have a solution for every patient, but what we are saying is we really try to understand in depth what's happening, and then try to determine the way to go as good as possible and as adjusted. And my dream is that we recognize the role of the immune system in glioblastoma. There is so much evidence now that there are immune cells before the treatment starts, but they are basically killed with radiation and chemotherapy. So once we understand the disease and once we recognize the potential the immune therapy has, then we try to build the immune system of a patient, but then this has to be supplemented by additional treatment in order to help the immune system to function properly. So my hope and vision is that we get away from the brutal strategies to actually not being able to cure this disease, to a way where I'm not saying we have a cure, but we try to control the disease in the most precise way possible with the highest possible quality of life for a patient.